Hello students, now we will start with new topic, that is reverse transcription. Reverse transcription means synthesis of a DNA from a RNA. We know that synthesis of DNA from one existing DNA, this process is called as autocatalysis or DNA replication. Another thing, synthesis of RNA from a DNA, this process is called as heterocatalysis or transcription. In the same way, synthesis of a DNA from an RNA, this is called as reverse transcription. Reverse transcription first observed by Temin and Baltimore. In the year 1979. This reverse transcription, also called as Teminism. Many number of or types of organisms are having DNA as genetic material, but there are some types of organisms that have RNA as genetic material. Example for such organisms is retroviruses. Retroviruses have RNA as genetic material. So, for their survival, when they infect a host, they synthesize a new DNA that is related to their gene sequence and that can synthesize the proteins and the enzymes or the viral particles according to their body structure. So here, one common point we will see, synthesis of DNA, synthesis of RNA, that is mRNA from a DNA, and synthesis of proteins or translation of proteins from the genetic information of mRNA, total we are calling as, the process is called as central dogma. But here, this RNA is again getting converted into DNA by the process of reverse transcription. For this process, it needs some of the enzymes called as RNA-dependent DNA polymerase. In transcription, we have seen the existence of RNA polymerase 2. But here we are seeing RNA-dependent DNA polymerase, which is also called as reverse transcriptase. This is also called as RNase. Now, when an RNA is existed inside the retroviruses, it is having the coordination or assistance of another type of special enzyme, that is reverse transcriptase. For suppose, if we think this is a retrovirus, it is having genetic material as RNA and it is also having another enzyme very near to it. Always these both are transported into the host organism and do the process called reverse transcription. So this is called as reverse transcriptase. RNA containing retrovirus also has special type of enzyme that is called as integrase. This enzyme is called as integrase. So now we will see what are the functions done by reverse transcriptase or integrase. For suppose, think this is an eukaryotic host cell. And it will be having a nucleus. Inside the nucleus will be the genetic material along with nucleases. Nucleolus. So this will be nucleolus. This will be chromatin material. Now, a retrovirus will be infecting into the host. So during that infection, it incorporates its genetic material into the host along with reverse transcriptase. So this is reverse transcriptase enzyme. This is viral RNA. Now, this RNA enters into the host cell and it will synthesize a DNA which is complementary according to the gene sequence of RNA of the retrovirus, and that DNA is called as complementary DNA. If you think this is the RNA of the retrovirus and this is reverse transcriptase enzyme, this will be the newly synthesized cDNA. Now, one point regarding cDNA. cDNA has long terminal ends. To get converted into double-stranded DNA to transcribe mRNA. When an RNA has synthesized a cDNA, together we call as RNA-DNA hybrid. After formation of RNA-DNA hybrid, then the reverse transcriptase will degrade the existing RNA. So this will not exist anymore after the formation of RNA-DNA hybrid and also after the formation of cDNA, which is coded by reverse transcriptase enzyme. So that point also I'll write here. After formation of cDNA, the reverse transcriptase will degrade the viral RNA Now, this cDNA will be converted into double-stranded DNA. Now, the enzyme integrase now helps the cDNA, which is now double-stranded, to get incorporated into the host DNA. So, if this is the DNA of the host, now this viral DNA, which is synthesized in the presence of reverse transcriptase, now gets entered into the nucleus and gets incorporated along with the host DNA, like this. Now it will undergo the DNA replication and it will synthesize many number of fragments at the same time. Now here it synthesizes mRNA, which is similar to the gene sequence of the viral RNA, mRNA for virus. Now mRNA will come out of the nucleus and it will get dispersed into the cytoplasm of the host cell. Here it will do the process called as translation and synthesize the proteins that are required for synthesis of viral particles and also the enzymes that are required for synthesis of viral particles, and also proteins and whatever enzymes that are required. Now, these are responsible, both are responsible for synthesis of viral particles, again RNA and the reverse transcriptase, because here we have seen not only the proteins that are required for the protein coat of the virus, but also the enzymes like reverse transcriptase, integrase, etc. So, in this way, the viral particles along with the RNA of the virus and also the enzymes that are required for the virus are synthesized by the process of reverse transcriptase. So, this is about reverse transcriptase, which is discovered by Temin and Baltimore.